Hi, everyone. Welcome to today's edition of the H3A BioNet webinar. I'm just going to give a few minutes, 2 more minutes for people to join, and then we can get started. Thank you. Okay, I think we can get started. Welcome to the H3A BioNet webinar series. Today we have Dr. Etienne Patin, who will be talking to us about the demographic and adaptive history of Central African hunter-gatherers and farmers. My name is Chinakaho, and I'll be moderating today's session. Welcome, Dr. Patin, and thank you for availing yourself to be a part of the H3A BioNet webinar series and contribute. And welcome to everyone. Thank you for joining us. Just a few things. Yes?
Go ahead.
All right, thank you. Just a few things before we get started. We will be streaming this to Facebook Live, so the webinar is being recorded. The recording will be made available a day or two after this webinar. I'll post the link to the YouTube channel where you can access the recordings. And please remain muted and turn off your camera if you're not presenting. And yeah, I hope you enjoy the talk. And Dr. Patin, when you're ready, you can get started. Thank you.
Thank you very much, Chinakawa. So it's a great pleasure and honor for me to be here today. As Chinakawa said, my name is Etienne Patin. I'm a CNRS researcher at Institut Pasteur in Paris, France. And so today I would like to give you a talk about some of the results we obtained recently about the, let's say, population genetics of populations living today in Western Central Africa. And so I will organize my talk in two different parts. The first part will relate to the demographic history of Central African hunter-gatherers and farmers, and the other part about the adaptive history. I will get into the details after. Sorry. Oops, okay. So we are all familiar with this kind of figures that represent population movements around the globe. These figures are very popular, and most of them rely on population genetics studies. And I think it's quite interesting to realize here in the figure that usually no arrows are represented in sub-Saharan Africa, because most of these figures consider that most of the interesting population movements occurred outside of Africa. I think it's a problem and a limit, and based on the different studies I'm going to talk about today, our general aim is to learn more about the peopling history of sub-Saharan Africa, as well as how human populations living there adapted to the vast and contrasted ecosystems that are present in these regions. So we focus particularly on one important region of sub-Saharan Africa, the equatorial rainforest, for several reasons. The first one is that we know relatively little about the early human occupation of that region. And so we believe that using a population genomic approach could help learning more about the early human occupation of the region in a situation where there is limited archaeological records, because the region is characterized by acidic soils where in general human remains are not available. So a second important question that we would like to answer by studying this specific region is how the rainforest has impacted human demography. Does the rainforest can populations in the rainforest increase in size, for example? Do we observe signals of population expansion in populations who traditionally live in this setting? And the last question is about how human populations have genetically adapted to this rainforest environment, which is relatively specific in terms of, for example, humidity, but also exposure to pathogens. So broadly speaking, the region is inhabited by two general groups of human populations that I would classify into, on one hand, rainforest hunter-gatherers, that I would call RHG for the rest of the talk, for simplicity. So RHG correspond to actually the worldwide largest group of active hunter-gatherers on Earth. So that's also another reason why the region is particularly interesting to study, because it's not easy to study populations who are still active hunter-gatherers and to know how lifestyle, for example, has some relationship with genetic diversity, for example. So rainforest hunter-gatherers are generally characterized by a lifestyle that relies traditionally on hunting and gathering in the rainforest. Usually, rainforest hunter-gatherers live as small mobile groups, and most of them share distinctive phenotypic traits such as low stature. At the genetic level, we knew from early studies based on mtDNA and Y chromosome data that there is a substantial genetic differentiation between RHG populations and other populations, non-RHG populations. Also, there is some genetic differentiation among rainforest hunter-gatherers. And historically, these groups, RHG groups, have been separated into two general groups: the ones that live in the western part of the rainforest, represented in orange on the top on the plot on the top. These populations include, for example, the Baka, the Aka, the Bongo, the Bakoya, the Bakola, or the Betsan. And also the eastern rainforest hunter-gatherers, represented in green here, such as the Asua, Sua, NFA, who are usually called Mbuti in genetic studies, but also the Batwa or the Bakwa. So our initial question was to learn more about the history of these populations and also to learn more about the other group that is living in the same regions, that I will call in general Agre, for Bantu-speaking farmers. So these populations are characterized by a sedentary lifestyle, and they are usually practicing agriculture, such as agriculture of yam, millet, or sorghum. An interesting aspect of the history of these populations is that they are characterized by the relatively low linguistic diversity. This relatively low linguistic diversity was interpreted as evidence of a recent linguistic expansion of the so-called Bantu languages starting around 5,000 years ago, from the frontier of present-day Nigeria and Cameroon. And it was also suggested that this linguistic expansion was accompanied by the emergence and the spread of agriculture. At the genetic level, we know from early studies that there is relatively limited genetic differentiation among agri groups. Okay, so in this first part, I'm going to detail all the results we obtained about the demographic history of these populations, Central African hunter-gatherers and farmers. The general questions we wanted to address were related to the general peopling history of the region. When did these groups diverge from each other, for example, and how did they interact in the more recent history? So to do that, we used the genetic data, and we infer demographic parameters of these populations using a variety of methods. In this case, I'm showing you a model that we estimated based on the exomes of 300 individuals of Western and Eastern rainforest hunter-gatherers and farmers. So we obtained this model by searching for a demographic model that best fits the observed data using simulation-based composite likelihood methods, in this case, FAST-SYNCO2. So when we do that, we find a number of interesting results. The first one is that we estimate the separation between rainforest hunter-gatherers, RHG, and Agri at around more than 100,000 years ago. So these results really supports relatively deep population structure in Africa. So it's actually not the earliest divergence state that has been estimated among sub-Saharan African populations, which is actually estimated to around 200,000 years ago for Khoisan populations from Southern Africa. But still, we show here that also populations that traditionally live in the African rainforest have separated from other populations relatively early in time during the later Pleistocene period. We also find that rainforest hunter-gatherers from the western part and the eastern part of the equatorial rainforest share common recent ancestors. They separated from each other around 20,000 years ago only, which corresponds more or less to the last glacial maximum at the time period where the rainforest was divided into two different refugia. We also find that relatively clearly rainforest hunter-gatherers have experienced a five-fold reduction in population size, here represented by the width of the squares, while ancestors of Agri, of Bantu-speaking farmers, experienced a 2- to 3-fold increase in population size. We suggest this is related to their respective lifestyles. There are two important observations that we can also make from these results. The first one is that we date the expansion in Agri populations at around 10,000 years ago, which predates the emergence of agriculture in the region. This is a bit contradictory to the general hypothesis that agriculture allowed population expansion. Here we tend to suggest that this is the other way around. The other important observation based on these results is that the population reduction of rainforest hunter-gatherers is also relatively recent in evolutionary times, which suggests that for a long time period, ancestors of rainforest hunter-gatherers were corresponding to a large structured population, and there today are less populations of rainforest hunter-gatherers today are smaller. Finally, based on the same data, we also find strong evidence for large migration rates from agri populations, farmers, to rainforest hunter-gatherers in the last 1,000 years, which is possibly due to the strong socio-economic interdependency that we observe today between these populations. It's also interesting and reassuring to notice that we found approximately the same model around more than 14 years ago, based on only 300 Sanger sequence SNPs and not whole exomes. It's actually a little bit depressing because I spent 3 years to build this model like 14 years ago, and we spent like 2 or 3 years to build exactly the same model with much more data. But anyway, it's actually even more depressing to notice that the same model was found by Luca Cavalli-Sforza and colleagues in 1969 based on only 14 blood group antigens. So that's actually a small tribute to the great work of Luca Cavalli-Sforza, who passed away some years ago. It's actually considered as the father of human population genomics. Okay, so we found that our RHG and AGR have different demographic histories. On one hand, RHG experienced population collapses recently, while farmers experience expansions. So we wanted to know if these different demographic histories have impacted selection efficacy in these populations. So what do I mean by that? We know from population genetics theory that populations that are small in size experience more genetic drift, where allele mutations can fluctuate in frequency like randomly, stochastically. And because of genetic drift, in some cases, deleterious mutations, and in some cases, and mostly recessive deleterious mutations, can reach high frequency in populations that are small in size. And so we wondered if the demographic history of rainforest hunter-gatherers, and in particular their recent population collapses, have decreased selection efficacy in these populations, which could result in these groups into a higher prevalence, for example, of recessive genetic diseases. So to do that, we estimated what we call the genetic load of individuals from different populations, and we compared them across populations. And we did that from our demographic model here. So we simulate mutations under negative selection under this demographic model. We compute an estimation of the load, and we compare these estimations across populations. So we did that assuming 2 different genetic models: the additive model, where the heterozygotes have an intermediate phenotype compared to homozygotes, and the recessive model, where heterozygotes have the same phenotype as one of the 2 homozygotes. So when we concentrate on the plot on the left, we look at the additive load on the y-axis and time on the x-axis. And so what we observe is that we see no deviations in load in any of the populations represented by lines in different colors when we concentrate on the additive load. So that's in line with previous studies showing that in general, additive load is the same across human populations, whatever the demographic history they experienced. On the right, conversely, we do find differences in the recessive load of populations. If we take, for example, Europeans in blue that we also analyzed as some kind of a control in our study, we do find that at the time of the out of Africa bottleneck, the genetic recessive load increases quite a lot, which is expected because they experience more drift. And some of the deleterious mutations that segregating these populations at that time can increase in frequency. But then they also experienced strong expansion, which reduced drastically their recessive load. Now we should focus on the populations from Western Central Africa. In red, we have farmers, and in green, we have the rainforest hunter-gatherers. We do find that their contrasted demographic histories in the last 1,000 years should result in different recessive loads based on simulation data. However, we also find that gene flow, which is simulated in our demographic models, also reduces the load in the rainforest hunter-gatherers in green, which is something then that we wanted to confirm based on the observed data. So what do we observe now if we look at the data itself, because these were simulations of deleterious mutations? What we did is to estimate the mutation load in different populations, and we computed population ratios of these loads. For example, on the lower row here, you have the ratio between the load in Western hunter-gatherers and Western Bantu-speaking farmers. So what we see actually is that this ratio does not significantly deviate from 1, which means that in general, rainforest hunter-gatherers do not have a higher additive or recessive load at mutations that are suspected to be deleterious because they have a high GERB score. And so these results, together with simulations, tend to suggest that actually gene flow from AGR through RAG has counteracted the predicted higher recessive load that was expected in hunter-gatherers because of their recent population collapse. Okay, so we talked a little bit about RAG. Now let's talk a bit about AGR. So most of the AGR populations I'm referring to are actually Bantu speakers, and it has been, as I said previously, suggested that Bantu languages expanded across the entire sub-Saharan African continent around 5,000 to 3,000 years ago. But there are still important questions about these expansions, and the first one is were these Bantu expansions endemic or not? So what do I mean by that? I mean that there are basically two competing hypotheses to explain the fact that most Bantu languages are very close to each other while they are spoken in very different places in sub-Saharan Africa. The first hypothesis is that Bantu languages were transmitted from population to population, but populations did not move. This is what we call, in this case, the cultural diffusion hypothesis. Populations stay and languages move. Alternatively, we could also imagine that people moved with their languages. So this is what we call the endemic diffusion hypothesis, where actually people move with their language. So in the case of the Bantu expansions, we could test that by simply checking how genetically diverse are Bantu speaking populations. If they show a very low diversity, that probably means that they share very recent common ancestors and they separated from each other in the last thousands of years. Also, we would like to ask if Bantu-speaking people are mixed with local populations, if the Bantu expansion was endemic. And if, again, the Bantu expansion was endemic, what were exactly the early roots of these expansions in our region of interest, that means Western Central Africa. So to answer these different questions, we studied, sorry, I'm going to skip that. We studied the genetic diversity of around 1,300 individuals who speak Bantu languages. And we merged these data with reference populations from other places in sub-Saharan Africa so that we could learn more about their genetic interactions. So we, in this case, conducted a classic analysis in population genetics, which is an admixture analysis where we cluster individuals according to a predefined number of clusters. And we also express estimate the admixture proportions for each individual from these different clusters. Okay, so when we do that without Bantu-speaking populations, we actually observe well-known populations in the genetic landscape of sub-Saharan Africa, including Western African ancestry in gray, Eastern African ancestry in general in pink, and also ancestry from rainforest hunter-gatherers from the West and the East in green, and ancestry from click-speaking populations that are usually hunter-gatherers, such as the Khoisan, and also hunter-gatherers from East Africa in pink, sorry, in yellow and blue colors. Now we would like to see how are the Bantu-speaking populations on the top of this picture. And so we sampled a lot of populations close to do that, we sampled a lot of populations from close to the postulated homeland of Bantu expansions at the boundary between present-day Nigeria and Cameroon. And what we observed is that all Bantu-speaking populations clustered together, they all share the same red color that corresponds to a unique cluster, genetic cluster, when doing the admixture analysis. So this usually suggests, in general, that they really share similar genetic profiles. Also, when we estimate some indices that are used to measure the degree of genetic differentiation between populations, namely FST, we find very low FST values, which again suggest little genetic differentiation and structure among Bantu-speaking farmers. So globally, these results are in line with the Genomic hypothesis. That means that the Bantu languages were diffused together with populations. Actually, populations migrated with their languages. Based on the same results, we could also find that evidence for genetic admixture between Bantu-speaking farmers and local populations. We could discern three different situations that are of interest. The first is in South Africa, so it's well known that there has been a mixture between Bantu-speaking first-comers, and local autochthonous populations such as the Khoisan. What we found interesting is that there is not a large period of time between archaeological evidence for Bantu migrations in South Africa and the admixture dates that we can obtain from the genetic data. There are like some 100 years between the two dates, which suggest that the Bantu migrations were followed relatively rapidly by admixture in this region. But that's not the case everywhere. When we then look at Malawi, for example, and these results are not coming from our study, but from a study by Pontus Skoglund, the situation is slightly different, because they actually observed that in the genomes of present-day Malawi populations, there are no genetic traces of populations who lived there prior to Bantu expansions. This suggests that here there has been a strong population turnover and not extensive admixture. And the third case, which we are interested in particular, because it's occurring here in Western Central Africa. So in this case, we know that there is archaeological evidence for early arrival of Bantu migrations in the region, because this is close to the homeland of postulated homeland of Bantu expansions. So around more than like 3,000 years ago. But in clear contrast with South Africa, we date admixture with local populations at around 800 years ago, which is very different from the 3,000 years ago estimated by the archaeological record. So this suggests for us that there has been, in contrast to South Africa, strong barriers, cultural barriers against intermarriages between populations of Agr, Bantu-speaking farmers, and rainforest hunter-gatherers who traditionally live in the region. By the way, there is also another way to interpret these results, the fact that Bantu speakers arrived like 3,000 years, but admixed only 800 years ago with local populations. And this is possibly because there has been actually two different waves of Bantu migrations. The first one 3,000 years ago, which did not result in admixture with local populations, and the second wave that occurred around 1,000 years ago, which in this case resulted in admixture. We next wanted to learn more about the early roots of Bantu expansions, and we used again the genetic data to learn more about these early roots. So how did we do that? So what we did is to look closely at the genetic relations between, in this case, Bantu-speaking farmers from East Africa. This is the Bakiga from Uganda. And we tried to find if the part of their genome that is coming from Bantu-speaking farmers is closer to people living today on the southern part of this region, on the northern part of this region. So why did we do that? This is because there are actually two competing hypotheses to explain Bantu expansions. The first hypothesis is what we call the early split model, where Bantu-speaking farmers first separated very early, close to the homeland of Bantu expansions. And so East Bantu-speaking groups should be equally genetically distant to people living in the southwest part of Africa. And alternatively, the other model that was proposed is the late split model, where Bantu-speaking farmers migrated first southward and then separated after they crossed the rainforest. So to test these two competing hypotheses, we simply checked if the part of East Bantu-speaking populations is more related to northern populations or southern populations from the Western Central African region. So what we found is actually represented by the width of these circles. And so we find that the large majority of the ancestry coming from Western Central Africans in the Bakiga, Bantu-speaking populations from the east, is very close to Angolans and not very close to Cameroonese people, for example. So this is definitely in line with the late split model, which suggests that Bantu-speaking populations first migrated southward and then migrated eastward. We find similar results when focusing on populations from South Africa, Bantu-speaking population from South Africa, Lesotho. And here again, we find that some of their ancestry is deriving from the Khoisan, as already known, but all their Western Central African ancestry is much closer to Angolans than Cameroonese, again in line with the late split model. So these results tend to suggest that Bantu-speaking farmers migrated first southward through the forest or deforested corridors, and then migrated eastward and southward. Using the same data, we also wanted to ask a question that is more related to the transatlantic slave trades. we merged our unique data of Western Central Africans with the genomic data for 5,000 African Americans from the US. And the basic question we wanted to answer is, can we find evidence that there has been a genetic contribution of Western Central Africans to the transatlantic slave trades? Indeed, most of previous studies assume that the vast majority of the African ancestry of African Americans is coming from Benin and Nigeria, represented usually by the Yoruba population. So using our genetic data, we used the same kind of approaches as before. We wanted to estimate what is the relative contribution of different source African populations to the African genomes of African Americans. And the values are represented here in blue. So we indeed confirm that the majority of the African genome of African Americans is closer to populations such as the Yoruba of Benin and Nigeria. But we also find 30% of their African ancestry coming from Western Central Africa, which was relatively a novel finding for genetic studies, but definitely not a novel result for historians, because based on historical records, it's already known that a number of the slaves who were transported to North America were coming from Angola in particular. Interestingly, we also find in the African genomes of African Americans around 5% of rainforest hunter-gatherer ancestry, represented in green here, which is again in favor of a genetic contribution of Western South Africans, because these are the only populations among these ones who have large amounts of rainforest hunter-gatherer ancestry. And actually, it also fits very well with our results, because 30% times 16% is around 5%. So that's what we expect. Okay, just to conclude on this part, I should just check the time. Okay. We conclude from these results that Central Africa has probably hosted one of the largest groups of rainforest hunter-gatherers who separated more than 100,000 years ago from other human populations. I want to highlight here that we don't know very well the relationship of rainforest hunter-gatherers with Khoisan hunter-gatherers from South Africa. And this needs to be resolved by genetic studies. We also find that hunter-gatherers and farmers from Western Central Africa have experienced contrasted demographic histories, but these different histories did not result in different burdens of deleterious variants. And we suggest that this is because gene flow from AG to RG has counteracted the recessive load that is due to the recent population collapse experienced by hunter-gatherers. We also conclude from the genetic data that endemic Bantu expansions first moved southward from the Nigeria-Cameroon frontier through the equatorial forest, and that these that Bantu-speaking populations have often been admixed, with local populations. Finally, we also find that African Americans descend from people transported from both Western and Central Western Africa in agreement with historical records. Okay, so now I skip to what I would call the adaptive history of these populations. Main questions being, at which extent human populations have adapted to the rainforest and the rainforest, African rainforest, sorry. So to do that, we conducted a study based on 600 exomes of populations, again, of rainforest hunter-gatherers and farmers. This work was mostly done by Marie Lopez, PhD student in our lab. So to do that, we search for what we call the genomic signatures of classic sweeps. A classic sweep is a model that assumes that a de novo mutation is advantageous, and because it is advantageous, it will increase in frequency in the population rapidly, And this will leave specific genomic signatures that we can detect with some summary statistics that we call neutrality statistics. So we used these neutrality statistics to scan the genomes of several rainforest hunter-gatherer populations to search for genes and mutations that have been advantageous in these groups. So we found two strong signals. A first signal of positive selection that was already reported by Sarah Tishkov and colleagues in a locus that includes two genes. One is involved in height, but the other is strongly involved in immunity. So we tend to suggest that it could be actually related to adaptation to pathogens that are particularly prevalent in the rainforest. And the second locus, which was described for the first time, is at the TRPS1 locus, a transcription factor that is involved in stature and craniofacial traits. This signal is quite strong, and it's also shared across all the rainforest hunter-gatherers populations that we investigated. So these results would suggest that maybe the low stature that is characterizing hunter-gatherers from the rainforest has been advantageous, has been adaptive. And so to test that more systematically, we search for evidence for what we call polygenic selection. In this case, we don't search for selection at a specific mutation or specific gene, but we would like to know if trait-associated alleles have increased under positive selection in one population and not in the other. So this is what we did, and the basic idea was to test if the differences in stature that we observe today between rainforest hunter-gatherers and farmers is due to selection, positive directional selection, and was selection occurring in rainforest hunter-gatherers or farmers. So we took all the mutations that are associated with height in Europeans, which is not perfect, but at least that's the best we can do. So we took GWAS SNPs associated with height in rainforest hunter-gatherers and farmers, and we verified if these mutations have larger values for neutrality statistics than random mutations. So the idea is that if trait-associated, height-associated, for example, mutations have higher neutrality statistics than random mutations, this would suggest that the trait has been under directional selection, has been advantageous in this group. So this is represented here by the circles, and significance is represented by the stars. And so the 5 first columns are rainforest hunter-gatherers, and the 2 last are Bantu-speaking farmers. So we do find some evidence, good evidence for directional selection, polygenic selection on height in the rainforest hunter-gatherers, but not in the farmers. We also find some evidence for other traits, such as H-at-min-arc or diastolic and systolic blood pressure. There is some caveat when you do this kind of analysis, because we know that the same mutations that are associated with, for example, height, are also associated with H-at-min-arc, for example. This is a case of pleiotropy. So if you find selection on one trait, such as height, you could find the same signal of selection on H-at-min-arc simply because the 2 traits are genetically correlated. They share some mutations associated with both traits. So to try to see if our signals are due to pleiotropy or not, with height mainly, what we did is simply to exclude from the data any mutations that are associated with height, and we lose all the signals that we observed for all the traits that are here, that are not height, basically. So that would suggest that there is strong positive selection on height, and the other signals are due to pleiotropy. We also did the other analysis, the complementary analysis, where in this case we keep SNPs associated with height, but we exclude any mutations associated with the traits that are listed here on the left. So when we exclude mutations associated with age at mean-arc, we still keep the signal with height. And this is also true for any trait that we investigated. So together, these results suggest that indeed there has been positive selection in the form of polygenic adaptation on height in rainforest hunter-gatherers. Sorry. Okay, so that's about rainforest hunter-gatherers, but we also wanted to learn more about the adaptive history of Bantu-speaking farmers. And so when we scanned the genomes of ag populations, we actually found two interesting signals that we were not expecting. Indeed, we found in the genomes of Bantu-speaking populations from the east a signal at the lactase gene. Why do I say it's relatively unexpected? This is because lactase persistence alleles are virtually absent from Bantu-speaking populations from the West, and they are observed at like almost 30% frequency in Bantu-speaking populations from the East. Actually, these two groups, East and West Bantu-speaking farmers, have diverged very recently since the Bantu expansions. So it's intriguing to see such a difference in allele frequency between the two groups, Western and Eastern Bantu-speaking farmers. And so to test if this observation is due to selection or not, in the form of post-admixture selection, we used new methods, and we indeed find a strong signal of selection since admixture in Bantu-speaking farmers from East Africa. This result means that Bantu-speaking farmers, as they arrived in East Africa, admixed with local populations, acquired these lactase persistence alleles from probably pastoralist populations who live there, such as the Maasai, for example. And then the mutation that they acquired by admixture increased in frequency very rapidly since admixture under strong positive selection. So this scenario that we usually call adaptive admixture is relatively novel in humans and has been described for the first time with these examples and other examples in Africans. So we also found the same kind of signals in Western Bantu-speaking farmers. They actually show a very clear excess of rainforest hunter-gatherer ancestry at the HLA locus. So it seems to suggest that Bantu-speaking farmers from the West, as they're mixed with rainforest hunter-gatherers, acquired some adaptive mutations in the MHC locus, which is known to be associated with a lot of infectious diseases, suggesting maybe that they acquired genetic resistance to some pathogens that are specific to the region. So overall, this suggests that ag populations rapidly acquired adaptive traits through admixture, gene flow, with locally adapted populations. We extended this idea to other populations, including populations that are descended from not transatlantic in this case, but oceanic slave trades, Indian Ocean slave trades, sorry. So we know that there has been also some slave trades on the eastern part of Africa, not organized by European settlers in this case, or colonists, but by Muslims living in the Saudi Arabia in general, and coming from the Omani Empire at that time. So slaves were usually transported from the coast of Tanzania and also Mozambique to Pakistan and also Iran and Iraq and India. So we studied a particular population that descends from these Indian Ocean slave trades. This population is called the Makrani from Pakistan. The Makrani have approximately 20% of their ancestry deriving from African populations. relating more or less to Mozambican populations, Bantu-speaking Mozambican populations. And the rest of the genome derives from local Pakistani populations, in particular the Baluch. So we did the same kind of approaches. We wanted to search for evidence for post-admixture selection because these populations are definitely admixed. And we actually found a strong signal of an excess of sub-Saharan African ancestry in the Makrani. We observed this signal at a locus that is well known. It's the ACR1 gene, which is responsible for the Duffy null allele, that is known to protect against Plasmodium vivax malaria. So this result is supported by the fact that there is 44% African ancestry at the locus, while the rest of the genome is only 17% of African ancestry in the Makrani. Okay, when doing simulations, we also find good evidence that there has been selection since admixture. And globally, these results tend to suggest that ancestors of Pakistani populations have inherited from African slaves this Duffy-null allele that has rapidly increased in frequency in the region. And we know today that Plasmodium vivax malaria is endemic in Pakistan and is actually responsible for most of the severe cases of malaria. So again, that suggests that admixture gene flow can facilitate genetic adaptation by promoting the spread of adaptive alleles from populations to populations. Okay, I think that's my last slide. We also wanted to learn more about a specific well-known example, iconic case of balancing selection. That is the sickle cell anemia mutation beta S at the hemoglobin locus. So this mutation is well known to protect against malaria and Plasmodium falciparum malaria in particular. So we wanted to learn more about the origin of this mutation in our data, because we know that this mutation is particularly frequent in West Africa, but also in Western Central Africa, particularly in the rainforest. It has been usually suggested that there is some kind of a link between the mutation and the rainforest, which raised the hypothesis for us that the mutation may have occurred in rainforest hunter-gatherers who traditionally live in the rainforest and was then transmitted to Bantu-speaking farmers at the time they are mixed together. And when we looked at the data in more details and we estimated by approximate Bayesian computation the age of the beta S mutation in farmers and hunter-gatherers separately, we found exactly the opposite of what we were expected. Actually, when you look at the plot on the left, you see the posterior probability distribution of the age of the sickle cell mutation in rainforest gatherers in blue and agri populations in red. And we see that the mutation has occurred approximately 24,000 years ago in ancestors of agri populations, while it's much more recent when we date it in rainforest hunter-gatherers. This suggests strongly a model where the mutation first occurred in agri populations and was then transmitted 6,000 years ago, or earlier, or more recently, sorry, to rainforest hunter-gatherers through gene flow. So again, that would suggest a case of adaptive admixture where an advantageous mutation, here mutation of the balancing selection, was occurred in one population and then was transmitted to another population through gene flow. Okay, to conclude with the second part, we find some evidence from the genomes of rainforest hunter-gatherers that human populations have indeed locally adapted to the rainforest, particularly at genes and mutations that affect stature and not age at menarche. Why do I mention age at menarche? Because some anthropologists suggested that the lower stature observed in rainforest hunter-gatherer was not due to selection for lower stature per se, but actually selection for an earlier age at menarche. So here we find some evidence that it's actually due to selection on stature itself. We also find that the geographic migrations and expansions of Bantu speakers were accompanied by the acquisition by Bantu speakers of adaptive mutations through gene flow from locally adapted mutations, which a scenario that we call adaptive admixture. And we find that malaria protective alleles have spread from Bantu-speaking populations to rainforest gatherers and Eurasian populations, and also in the other way around, supporting a key role of adaptive gene flow in human genetic adaptation. And to conclude, I would like to thank all the members of our group, Human Evolutionary Genetics at Institut Pasteur, in particular Luis Quintana Miosi, who is the head of the lab, and also PhD and postdocs, PhD students and postdocs who worked on this study, including Sebastian Cuadros Espinosa, Marie Lopez, Athanasios Kouzatanas, and also the work of Guillaume Laval about the sickle cell mutation, and also collaborators. And I would like also to thank you for your attention.
Thank you for that talk, Etienne. It was very insightful and quite an interesting summary of the work that you've been doing so far. I don't see any questions in the chat box, but the participants are welcome to either unmute their mics and ask their questions or raise their hands to ask the question. And you can also use the chat box to ask the question. Thank you.
Yeah, I have a question.
Please go ahead.
So I hear the speaker, saying West Africa, East Africa, and representing by a few samples that West Africa is just homogeneous, and few samples are representing the region. As the same thing, East Africa. So there is much diversity. I feel it's more of helicopter science, doing few samples and concluding for the entire continent or region. So is there any plan to expand the sampling, or, I feel it's more of based on convenient sampling right now. So do you think this research will hold if you have really in-depth sampling from each geographic or adapted to regions? So,
okay, thanks for this question. I think it's a very important question. So we definitely oversimplified the genetic diversity of populations from West Africa and East Africa in particular. I definitely agree. So that was the best we could do at that time, because most of our sampling efforts were concentrated on the region I was focusing on, which is Western Central Africa. So in this case, we sampled more than 1,500 individuals from mainly Cameroon and Gabon. And it was a little bit difficult for us as a single lab to also collect the same kind of sampling, to do the same kind of sampling efforts in other parts of Africa. But I think it's also highlighting really the importance of consortia such as H3 Africa, because one main caveat of population genetic studies in Africa is that each group is focusing on a given region, and we definitely need to have a broader picture of the genetic diversity of all populations. So yeah, that I would tend to say that this is clearly a limit of what we did. Then to answer specifically about the question, do I think that our results will hold with more populations? I do think so for most of our results, because most of our results relate actually to Bantu-speaking farmers and rainforest gatherers from Western Central Africa, which in this case, we have a lot of samples. We probably have the largest collection of rainforest hunter-gatherers from Western Central Africa. So for most of our results regarding the demographic model, regarding selection scans, I think it's fine. About the Bantu expansions, not sure, I must say. So because we indeed increased largely the sample size of Bantu-speaking populations in the genetic data. And also we focused on a key region, which is like very close to the homeland of Bantu expansions. But that's true that we are definitely missing a lot of data. And I can already tell you that collaborators, not myself, because I'm focusing more on Oceania now, but collaborators in particular Karina Schlebusch, they are conducting the same kind of analyses on larger samples and with much more populations from all over sub-Saharan Africa, and my conclusions are reproduced. So that's reassuring, but I definitely agree with you that this is a bit that our conclusions could be limited given the number of populations we studied outside of Western Central Africa.
Thank you for that. Is there another question? We'll stretch it for another 4 minutes and then conclude the call to allow for more questions.
Sorry, I have another question in terms of ancestry. When you say East African ancestry or West African ancestry, so what are you exactly saying? All are African ancestry, in my view. What are the unique feature of East African ancestry versus West African ancestry?
It's again a good question. So this is oversimplification. I'm a little bit obliged to get into these simplifications to make it simple, but actually I'm just showing colors and so on, but what do they mean exactly? So for example, when we see an excess of what I would call here by simplification East African ancestry represented in pink, adolactase gene in Bantu-speakers from the East Africa, it's actually in this case specifically Maasai ancestry that is close to Maasai populations. So as you understood, it's a very simplified picture. We can't say that Maasai are representative of all East Africans. Actually, East Africans are quite structured and they have a lot of diversity. So yeah, so that's just to make the story simple. But that doesn't mean this is wrong, right? So it's just we just need to be careful. For West Africans, most of the results here suggest that this ancestry is shared basically by populations from Senegal to Nigeria. Again, if we were to do a study specifically on Western Africans, we would definitely find structure among them. And in my case, I already know that my results correspond to ancestry that is most frequent in Yoruba populations, particularly from Benin and Nigeria.
Thank you, Etienne. I think we'll conclude the session there. Thank you for the questions and the answers as well to respond to those. I think for me, the most important thing I can take away from this session is that the work that you're doing is quite important in contributing towards social engineering, because a lot of people aren't aware of their ancestry or genetically and how genetically we're similar and how that influences where we find ourselves today. Like how you're saying that the admixture is linked to the Bantu-speaking languages that we see today. And I think that's important work that should be carried on. And as the person who asked, TM, who asked the questions, that the sample size should be representative of the populations and that they're talking about. Yeah, so I think that was a really insightful talk. Thank you very much. Thank you everyone for joining us. As I've said, the talk will be available on our YouTube channel. You can access it from there, and we hope to see you for the next webinar next time. Thank you, everyone.
Thanks a lot. Have a nice day.